Hi, everyone. Thank you for joining us today for this webinar organized in collaboration with REPROCELL. As Bob mentioned, so I'm Katia Lefsians, and I'm currently working as a product specialist for Cell Collapse, which is a biotech company based in Stockholm, in Sweden, specializing in the industrial GMP production for high-quality mesenchymal stromal cells. And during our time together, we will, of course, define the MSCs, what they are, and we will also present the multiple functions and more specifically, the action on the immune system. We will also look on their clinical application and the various studies carried out over the last 2 decades on this topic. And finally, I would like to introduce Cell Collapse, who we are and what is our current missions. But let's start with the MSCs and their definition. So the mesenchymal stromal cells, they are multipotent cells, as you know, meaning that under certain conditions, whether in vitro or in vivo, they can mature and differentiate into multiple specialized cells. The cells have the capacity to self-renewal, so they can divide while retaining their permanently undifferentiated state. So in this way, the pool of stem cells can be perpetuated throughout the life of the organism. And there is 2 main sources of MSCs. The first one are the adult MSCs that you can obtain from the bone marrow, from the fat tissue, so adipose, the dental pulp, and the whole blood, and the peripheral blood, I'm sorry. And you also have the neonatal tissue-derived MSCs that you can obtain from the extra embryonic tissue, such as the placenta, the amnion, and the umbilical cord. And currently, that's the adult bone marrow MSCs that are the most widely used in the different clinical trials that have been conducted or are ongoing on right now. And as multipotent cells, MSCs can produce a variety of cells limited to the germinal layer of origin, so limited to the mesoderm. So they can differentiate into bones, cartilage, they can differentiate into skin cells, cardiomyocytes, muscles, also in neurons and in hepatocytes, so all deriving from the mesoderm. Regarding the considerable therapeutic potential of these human multipotent mesenchymal stromal cells, they have generated an increasing interest in a wide variety of biomedical disciplines. But the investigators, they report studies of MSCs using different methods of isolation and of expansion, and so different approaches to characterizing the cells. This is increasingly difficult then to compare and to contrast the study outcomes, which impairs progress in that field. So to try to begin to address this issue, the Mesenchymal and Tissue Stem Cells Committee of the International Society for Cellular Therapy, also named the ICT, they proposed minimal criteria to actually define the human DSCs. I mean, what are the different criteria that cells should meet to be defined as MSCs? So the first one is that the cells, they must be plastic adherent when they're maintained in the standard culture conditions. The second is regarding the phenotype of the cells. So they must express the CD105, the CD73, and the CD90 markers. And we should find a lack of expression for the CD45, the CD34, CD14, and the class 2 HLA-DR. The third point is that these MSCs, they must be able into in vitro condition to differentiate to osteoblasts, adipocytes, and chondrocytes. And while all these criteria would probably require modification as new knowledge will unfold, we believe that this minimal set of standard criteria will facilitate a more uniform characterization of the MSCs and the exchange of data between the investigators in that field. So now let's focus on the biological function of the MSCs, because the cells are known for having a wide range of functions, but they are particularly well known for the immunomodulatory properties. Indeed, in numerous reports that you can find in the literature, they have shown that the adult MSCs, they can affect the immune T cells and B cells response. So they can suppress actually the T cells proliferation, their cytokine secretions, and the cytotoxicity effect of the T cells. And they can also regulate the balance between the Th1 and the Th2. They can also promote the generation of regulatory T cells and regulatory B cells. They may inhibit the B cells proliferation and stop their cell cycles. They can also affect the secretion of antibodies and the production of co-stimulatory molecules by the B cells. These MSCs, they can also inhibit the maturation, the activation, and the antigen presentation ability of the dendritic cells. And also, the other MSCs, they can inhibit the interleukin-2 induced natural killer cells activation. So all this part will lead like to a shutdown actually of the inflammation process that is triggered by this pool of cells. So also the underlying mechanism of the MSC's immunomodulation have yet to be elucidated, but they are likely mediated by both soluble factors that these MSCs are secreting, and that is named the secretome, and by the cell contact dependent mechanism in response to the immune cells. So more than a dozen of soluble factors are now known to be involved in the immunomodulation of MSCs, but let's just briefly talk about the most common one, the main one. So there is the transforming growth factor, the TGF-beta1, the prostaglandin E2, named PGE2, the hepatocyte growth factor, and also the indoylamine pyrrole 2,3 desoxygenase, that most known as IDO, the nitric oxide, and the interleukin-10. So in the literature, actually, the data showed that the pro-inflammatory cytokine, like TNF-alpha and interferon-gamma, now this is something really new, that if you put the cytokine, in contact with the MSCs, that will actually induce the secretion of IDO, PGE2, and IL-6 from these MSCs that will then mediate an immunosuppressive activity, and that will suppress the all different cells that we just talked about. But there is one point that remains unclear. It's what is the relationship between all these different soluble factors. But the effect of these soluble factors and the activity of the MSCs, we know now that it can be different depending on the origin of the MSCs, with the source of MSCs, and also the target cells and the microenvironment. So we thought that it's indisputable that MSC therapy contributes to immunosuppression, but further elucidation of the detail regarding the biological mechanism involved in this process is still required. There is also several reports on culture systems that have shown that the cell-cell contact is also a key factor involved in the immunomodulatory effect of the MSCs. There is a particular study from the hand groups that found that bone marrow MSCs not only decrease the survival and the proliferation of the T cells by contact-dependent mechanism, but they also increase the proportion of the Treg that has been produced. So just to summarize, in order to provide all this immunomodulatory function in response to the different stimulations, such as chemokine and that targets different immune cells, the MSCs are likely to employ both the direct contact and the soluble factors that work together for diverse and strong immunoregulations. Overall, you can see on the right that the MSCs also have a pro-regenerative ability, pro-generative capacity, because they promote a pro-generative microenvironment that promote the tissue local repair and regenerations. So the cells effectively participate in the tissue repair process through the immunomodulatory function that we just mentioned, but also through the inhibition, the apoptosis inhibition, the stimulation of angiogenesis, which is the stimulation of the generation of new blood vessels. They also have a neuroprotective ability, pro-neurogenerative functions, and in addition, pro-generative function, and also the MSCs play a central role during the wound healing process by coordinating between the local cells, the progenitor cells, and the cytokine and the chemokine, and also the extracellular matrix proteins. And under specific condition, the MSCs, so it has been shown for the bone marrow MSCs, they may directly or indirectly favor the generation and the proliferation of local progenitors, such as endothelial cells and fibroblasts. So that's why we say that they can't actually promote the stem cells growth and differentiations. So in addition of that, the MSCs, they are able to home to the sites of injury, where they secrete extracellular matrix components, they secrete chemokine and all the range of cytokines that we cite. They can, because also the MSCs home towards the tumors in a similar fashion way that they home to the injury sites, they can be, for example, useful for delivering cell-based therapeutic to tumor sites. And finally, the cells, they showed, as we just talked regarding the different criteria, they showed the ability to home and to differentiate. So in osteoblast, chondrocytes, and adipose cells, as we mentioned in the ICT criteria. So because of all the immunomodulatory potential that I just described, the clinical use of the MSC so far, it has focused mainly on patients that were suffering from autoimmune disease or chronic inflammatory pathologies. So for this patient, the intravenous injection of the MSCs has been shown to produce a real benefit because we actually found that there was suppression, that there was an effective suppression of the ongoing inflammatory process. But however, multiple studies have reported that there was a sequestration of the MSCs in the lung, actually, that after a couple hours after the infusion into the blood, there was a shortly clearance of the MSCs that we can track until into the lung, but after 4 or 6 hours, actually, you don't find any MSCs anywhere after the administrations. So the question that then raised is what is the fate actually and the biodistribution of the MSCs after the administration? What is the fate? But so this is something that has been actually studied a lot by different groups. And here I would like to highlight the work done by the Professor Katarina Leblanc. So she's a PI here in Sweden in Karolinska Institute, and she's actually a pioneer in the MSCs field because she's studying the MSCs since 20 years now, and she has conducted several clinical trials, and we are working in collaboration with her and her team here in Sweden because all the protocol that we are using in our company for the MSCs production, they are actually based on the work that has been done in her team. So she worked in collaboration with another scientist, Dr. Nadia Kadri, and they actually found that upon contact with the blood, the MSCs were opsonized by complement proteins, which will subsequently enhance the phagocytosis by the monocytes. So they're actually claiming that this result indicate that MSC's immunomodulatory effects that could be mediated by the monocytes that have phagocytized them. So actually by eating the MSCs, the monocyte could just then get the ability of some MSCs shift in their phenotyping and becoming immunosuppressive. But this is, of course, supposition, and there is still a lot of work going on that to try to actually better understand the complex mechanism that is governing the interaction of the MSCs with the different immune system components. But however, so far, what is clear regarding the and commonly accepted regarding the MSC therapy is that it's a safe therapy that is well tolerated by the patient. And there is now, you can find now several meta-analyses that has analyzed actually the result and the safety of dozens of clinical trials using the MSCs, or that is available in the literature. And I put it one on the slide. It's a meta-analysis dating from 2021. Which compiled the results of 62 randomized clinical trials involving more than 3,500 patients and covering around 20 types of disease. And so this patient, they also covering different routes of administration because regarding the disease, the patient was the MSCs were administered to the patient intravenously or by local injections. And so the result of this study and all the other showed that the administration of MSCs was safe in a different population compared with the placebo modalities. And that in conclusion, the MSC do not induce any allogenic response. There is no immunogenic effect. That's why in therapy, we can use actually allogenic MSCs. They do not induce tumors, and it's very well tolerated from the patient, as I said, because they didn't actually report any toxicity. I wanted now actually to talk more about some clinical study that has been performed using these MSCs, and more specifically about the pioneering work that was carried out by the Professor Katharina Leblanc, with whom we collaborate, as I said. So she conducted actually the first clinical trial using MSCs. It was bone marrow MSCs, the first clinical trial in Europe in the early 2000s to treat one patient. It was a child that was suffering from acute lymphoblastic leukemia, which is a rare type of cancer affecting the blood and the bone marrow, with the development of a large number of immature lymphocytes. And so this patient was treated with hematopoietic stem cells transplant. That was actually the gold standard treatment for the gold standard treatment for this cancer. But after the injection, this child developed an acute GVHD in the intestine and in its liver. So the GVHD, it's let's try to explain it easily. It's GVHD for graft versus host disease. It's the best known complication following the hematopoietic stem cells transplantation. So this GVHD develops when the donor T lymphocyte, the response to the antigen in the recipient tissue due to an HLA mismatch between donor and recipient. So the GVHD generally has a poor prognosis and few effective therapy are available. So usually people are using in the clinic steroids. But this patient therefore received allogeneic bone marrow MSCs 70 days after the hematopoietic stem cells transplantation. So yeah, first hematopoietic stem cell transplantation for treating the cancer, the leukemia. Then he developed a graft versus host disease, and so then he was treated by another injection, but this time adult allogeneic bone marrow MSCs. And the first point from that is that there were no adverse side effects recorded in this case for this patient. The second point is that the clinical response was remarkable. The patient actually recovered after 1 year. And the third point, this is actually what you can see on the figure shown here on the slides, that after the 2 injections, so the first injection was with 2 million cells per kilograms, and the second one with 1 million cells per kilograms. But after this 2 MSCs injection, you can see a drastically reduction in both the bilirubin level and the number of stools per day for the patient. And also it improved, it's not shown here, but it also improved the liver and the intestinal function of these patients. So thanks to the success of this case, I mean, this success was very encouraging for new perspective and for developing new control study of mesenchymal stem cells for the prophylaxis, but also for the treatment of GHD. So a new study, this time including 55 patients, all with acute GVHD, that were treated with allogeneic bone marrow MSCs, were published in 2008. So 55 patients, acute GVHD, and bone marrow MSCs infusion for treating that. So this clinical trial showed that the treatment actually did not have the same beneficial potential in all the patients treated. So among the 55 patients, they find that 30 patients were responding to the treatment. This is the patients that you can follow with the blue line on this graphs that I will explain just after. There is 9 partially responding and 16 non-responders patients, and you can show the follow-up of these patients that are represented by the red line. So as you can see from the graph on the left, the patient who responded completely to the treatment, so this patient, this one, they had a lower transplant-related mortality 1 year after MSC's infusion than the patient who actually responded partially or did not respond to the treatments. And regarding the overall survival of this patient on the right, the complete responders, this one, showed a higher overall survival 2 years after hematopoietic stem cells transplantation, which is unfortunately not the case of the half partially responder or non-responder that unfortunately, yeah, the last patient died after 2 years after the beginning of the hematopoietic stem cells transplantations. And so still regarding the GVHD, there were a third and more recent study that was carried out in the 2020 So as I say, again, on the treatment of the GVHD. So this time it's included 11 patients that were treated with repeated infusion of allogeneic bone marrow MSCs. And what was really interesting in this study is that it showed that the level of the chemokine CXCL9 and CXCL10, they can actually be considered as an early biomarker of responsiveness of the MSCs therapies. Indeed, as you can see, the CXCL9 and CXCL10 showed differential levels in responder that you can see here with the gray dot, and non-responder patient with the black dot, with actually a decrease in the measurement of the S2 chemokine in the responders after the multiple MSCs infusion. So after 1, 2, and after 6 infusion, you can see that there was strong decrease in this chemokine. So just for a short reminder, the CXL9 and CXL10 chemokine, they are mainly regulating the immune cells migration, differentiation, and activations. It regulates the diffusion of naive T cells to helper T cells, and it leads migration of human cells to the focal sites. So meaning that it's actually also aligned with the anti-inflammatory profile of the MSCs. So that was thanks to actually this very encouraging data on the graft versus host disease, the MSCs has been used in a wide range of clinical trial for treating many different diseases. And I wanted to talk about another one that was really interesting. It's about actually the ARDS. That is an acute respiratory distress syndrome. So for this pathology, the MSCs has been also used and infused to the patient. So the ARDS is characterized by, actually a severe inflammatory reaction of the lungs that is leading to respiratory failure for the patient. And it's still today a major cause of admission to intensive care unit worldwide with a high mortality rate, particularly for the severe case. So that's why in 2015, the author of the study, they successfully administered a single dose of donor-derived minimally expanded allogeneic bone marrow mesenchymal stromal cells to 2 patients. So these 2 patients were with severe ARDS, and they were both on the ECMO. So the ECMO is actually an ECMO for extracorporeal membrane oxygenation. So it's a machine that involves the pumping blood outside the body to a heart-lung machine that will remove the carbon dioxide and return oxygen-filled blood to the body's tissue. Actually, it replaces the lungs when they doesn't work. They just remove the dioxide carbon and return the oxygen to the patient. And so, as you can see on the slide, this is the chest X-ray that illustrates the progression of the acute respiratory distress syndrome for one of the 2 patients. So you can see that actually there is a progressive decrease in the pulmonary infiltrates, and it occurred like very early, because just 1 day after plus 1 day, it's meaning just 1 day after the MSC's infusion, you can still already start to see the decrease in the pulmonary infiltrates. And by the day 7, the chest X-ray findings had returned to the normal, and the patient was actually taken out from the ECMO the following day, so on day 8, and he was extubated on the day 12. And for the second patient, he also went out from the ECMO, but a little bit later, after 24 days, and he was also extubated on the day 41. So meaning that 2 out of 2 patients, they survived and they returned to work after 1 year, which is actually, very, very good and quite unexpected that to have this 100% survival rate, comparing with 40-75% for the current standard treatments that now that are given to this patient usually. So, and additionally, the authors, they actually provide a 5 years follow-up on these 2 patients, so still the same patients. So for assessing what was their health-related quality of life, their physical capacity, and the pulmonary morphology and function. So which all these different parameters was really good, which is unusual for the survival of ERDS. And so here you can see the patient 1, and you can see that this is the 5 years follow-up here, that there is only a mild pulmonary injury in the anterior part of the right upper lobe, here in the black and the white oval. And such a resolution are rarely seen in the long-term follow-up studies of conventional treated patients with ERDS. So it's very, very encouraging. And for the patient too, who had the most severe form of ERDS and was on ECMO support for 28 days before the MSC's infusion, this patient had no signs of pulmonary fibrosis 5 years after the MSC's treatment. So there is actually, on the strength of this promising result, there is a new clinical trial, again led by the Professor Catherine Blanc team, that is underway. This time, it's patients suffering from post-COVID infection ERDS, and they were treated with bone marrow MSCs as well. So initially, there were 11 patients that met the inclusion criteria that was set up for the clinical trial. But only 7 of them could actually be treated due to insufficient numbers of cells available. So this study is still ongoing on, so we don't have all the outcomes and all the data, but we can say that the results are encouraging because all the 7 patients treated actually survived. So we need to continue the follow-up for this. They need to continue the follow-up for the study. So we can see through all the different clinical trials that the MSCs, they are therefore hold great promise for revolutionizing the treatment of many serious diseases. But there are several difficulties that so far has limited the use of these cells more widely in the clinic. And the main thing is that there is a worldwide shortage of stem cells produced in the compliance with the good manufacturing practice. So there is a shortage of GMP MSCs actually. That can be due to the current production method, which are costly. Yeah, it's with the current method, it's very costly, very expensive to produce MSCs, and the yield is also like very low. So that's why, and above that, there is also another major challenge that is actually the lack of stability in the quality and efficiency of the different batches of MSC that are produced. So unfortunately, they don't all have the same reliable quality. So that's why, for trying to actually respond to that, Cell Collabs was founded in 2021. So to try to meet these challenges and to accelerate the research by making high quality MSCs available at a scale and to facilitate the future market approval of MSCs projects. So in our GMP ATMP approved facility, we produce MSCs on an industrial scale, which enable us to reduce the production cost, and we try actually to offer MSCs at a reasonable price. Also, regarding the quality of our MSCs, they are all actually coming from young, healthy donor that has been selected in accordance with the European regulation, and the cells actually, they are proceed in our facility that have been approved by the Swedish Medical Product Agency. So also, I mean, as I started to talk about it earlier, our company was founded on the research carried by the Professor Katharina Leblanc, that she has like now more than 2 decades of expertise in this field. And since our production protocol actually originated from the team, so we had like a technology transfer from her to our lab. So and to ensure the quality of the MSCs, we are also organizing a very strict and regular quality control of the cell that we are producing. So I also wanted to show you that actually we control the quality of the MSC that we produce at every stage of the process, from the donations to the GMP certified production and the transport, right up to the product delivery. And we are also providing help and advice to our partners regarding the reconstitution of the cell, because we are providing them actually a ready-to-use therapy. So we're explaining them how to thaw, how to reconstitute, and also advising them how to inject and delivering the product to the patients. So regarding the MSCs that we are producing, we are offering 2 types of MSCs. We offer MSCs for the research, so the research grade, and GMPs MSCs that are will be more dedicated, of course, for the clinical use. Regarding the information of this product, the information are similar. There is only two differences, being the number of cells per vial, because for GMP, of course, we can adjust the amount of cells per vial per dose regarding the number of patients that should be treated. They're all coming from bone marrow, from healthy volunteers between 18 and 30. Now it's more 28, actually. They are xeno-free. And the passage is also something different, because for the research grade, we provide passage 2 or passage 3. But for the GMP, they are passage 3. And also for the shipment, the preservation, it's into dry ice for the research grades, but liquid nitrogen for the GMP MSCs. So I also wanted to highlight that now we were able to elaborate, to set up many collaborations all around the world with different partners. So we are supporting both hospital and both companies by providing them our MSCs. So this is GMP MSCs. So we are working with Denmark hospitals, where there is an ongoing phase 1 clinical trial for the treatment of the Sjögren's syndrome. So actually, this is a syndrome where people has issue with the salivary glands. Actually, they don't produce enough saliva, so which is actually this issue is called xerostomia. And we are treating this patient with, they are treating this patient with our MSCs to try to regenerate the new glands. So this is more for the regenerative property of the cells. There is also another clinical trial in Sweden, phase 1 clinical trial for the treatment of hip osteoarthritis. Another one in Hong Kong, still a phase 1 clinical trial, this one for the spinal cord injury. And in the UAE, our MSCs are approved by the local authority, named the Department of Health. And our local partner is rated as the best multi-specialty hospital in the UAE. So, but as I said, we also have collaboration with companies. So here in Europe, in Ireland, for phase 1 clinical trial for cardiovascular disease. And in Italy also, still a phase 1 clinical trial, and also on the osteoarthritis. So just to try to make a summary of our company and what we offer. So we claim we are happy to offer actually high quality and affordable MSCs for both research and clinical trials. Our expertise is really based on a solid scientific background and all the research that has been done in the stem cells field. We are producing GMP MSCs, but we have flexible, and we can actually adapt the scale to support our customers in their different need regarding the clinical trials that they want to perform. And we are, of course, working in collaboration. We are offering collaboration opportunities with affiliated scientists. We are always happy to talk more about our product and to provide MSCs for treating patients that today, unfortunately, face to disease with treatment that are not as effective. So thank you very much for your attention, and I will be really happy to answer to any of your questions. Thank you.